you so much. Thanks for the invitation to speak to you all today. I think Dr. Mills, it's probably more overlap than I would have anticipated. I'd like to introduce a lot of the concepts that'll be very important that her work really underscored. So as a brief outline, very briefly for this audience, just talk about the kinome, our perspectives on some challenges and kinase target therapy in these rare pediatric tumor predisposition syndromes, NF1 and NF2, and I'll give a couple of vignettes on kind of where we've gone. And I'm a basic scientist, so really a lot of this is dependent on our interactions with my clinical collaborators here, and they really began when I was training with Gary at UNC with Wade Clapp here at Indiana. So very frequently, and this can be very brief, there are kinase targets that are very obvious, right? Drivers or really obvious evidence that they're implicated in disease settings. And I'm going to be talking mainly about tumor biology, cancer biology, and we've got kinase inhibitors for them. And as Dr. Mills underscored, even what we would, this is a BRAF V600E inhibitor. Now we know it's very important to think about the other things we're hitting, even with something that we view as pretty selective and that's even approved and in use in patient populations. And just to further underscore that, you're probably all aware of this, that there's now, I believe the number is 74, but since the first small molecule Gleevec or imatinib was approved for BCR-ABL, the Philadelphia chromosome, chronic myeloid leukemia, just numbers continue, and I think this trend is going to continue all the way that those that kind of reach the clinic because of the importance of kinases and cell signaling and how frequently it's dysregulated in cancer and other diseases, of course. So the challenge is kind of from my perspective is, what do you do when you don't have an overt or obvious kinase to target like BCR-ABL or BRAF? And I'm talking about NF1 and NF2 are both tumor suppressors, so they're marked by the absence of a protein. And although we know a lot about their pathway involvement, what they dysregulate, it still requires some kind of deeper study to figure out what are the best kinases we should be trying to hit. As Gary's lab has shown, Neil Rosen, Roger Lowe, and Levi Garraway, a number of other, too many to name, we now know in the clinic, on the preclinical side, tumors can rapidly adapt to treatment, even with these great targeted therapies. Even if we talk about, instead of an orthosteric inhibitor, an allosteric inhibitor that's much more selective, tumors can still adapt. And so I think looking at having an understanding of that, how the kinase signaling network changes, is very important as we think about combination therapies, drug holidays, even bringing in maybe immune checkpoint therapy as a combination for a lot of these things that we're studying. And last, Dr. Mills introduced this nicely, but we think about this too. Don't do this as deeply, develop these powerful databases that her lab is doing and the Sorger and Kuster labs have done, but we refer to them, we're very interested in what these so-called targeted drugs are really hitting. Often, for example, like dasatinib approved for CML hits 50 kinases. And I think Dr. Mills really underscored how frequently that happens for approved inhibitors. So when I was in Gary's lab now, 8 or 10 years ago, he had just published in one of those arenas where there's not an obvious kinase target, triple negative breast cancer. There's a desperate need to kind of find some good therapeutic approaches. And his lab had been building on, work from Henrik Daub, Matthias Mann, and Banchep and Kuster, and using type 1 or ATP competitive orthosteric kinase inhibitors, designing synthetic linkers that could adapt them so they could be coupled to cephorose bead to have an affinity matrix to capture kinases. We knew at that point, RNA sequencing, DNA sequencing, we can catalog mutations, we can look at the level of the transcriptome. You can even do total proteome, but Gary's lab was really interested in kind of using this to add additional intentional bias to the assay to try and capture kinases more selectively in their active conformation. So using an ATP competitive inhibitor and kind of adding that as part of the enrichment, as you can see in lysate from cells or tumor tissue from either mouse model or patient. And I think Gary's lab and a number of other labs kind of eventually found out that working on this independently really evolved to what ended up being a mixture. I think they trademarked the term kino beads at Cellzone, but Gary's lab multiplex inhibitor beads, but 4 to 6 kinase inhibitor beads that actually are dirtier. Here the intention is to get something that's pretty broadly selective to get the most comprehensive coverage of the kinome. And now that includes several hundred members. get that capture, and then be able to compare two conditions, treated versus control, as I'm going to show in a moment, even maybe genetic stages or stages of tumor progression, to give us insights into not only the effect of a drug, but also what kinases should we be thinking about targeting. And as many of you are probably familiar with, but once you've got that capture, then traditional, triptych peptide analysis or traditional methodology, this has really been aided by advances in mass spectrometry, increased sensitivity, now with the Orbitrap and ESI, electrospray ionization, to let us use lower and lower amounts. We're still kind of limited because we need to do an affinity capture. So we get to a certain point where if the amount is too low, but while I was in Gary's lab, we got this down to being able to use the amount of protein we could get, for example, from an 18-gauge needle biopsy. So could really work with our clinical partners to analyze patient specimens. And so then we could plot in this volcano plot kind of the response of the kinome. And with cell lines, we're talking about, several, a few hundred, 200, 250 maybe kinases. And that's in a cell line mixture. If we use a tumor, or there are more cell types involved, that number with where we stand now, probably getting into the 350 kinases that we're able to identify and quantify. So really powerful assay, giving us incredible insight into what we term the functional kinome. And so it's really based on a bias toward its activity state, but also its expression level and affinity for these beads. And so how have we been applying this to NF1 and NF2? I was inclined to do this in kind of numerical order of the two syndromes, but we actually historically were first contacted by Wade Clapp here at Indiana, where I am now, when I was still in Gary's lab, working on NF2 tumorigenesis, this schwannomatosis. And to give you a brief bit of background on this, this is actually a benign tumor syndrome. So it has a autosomal dominant inheritance pattern, really rare, affects 1 in 25 to 30,000 individuals. And they can either inherit from one of their parents or have a kind of a sporadic germline, or they can actually be a mosaic, a mutation or a hit in one copy of NF2, a tumor suppressor gene that encodes a protein called merlin. I'll talk about more in a moment. And this really pops up in the pediatric population. So Wade's the chair of pediatrics here. And by the time, many of these patients, by the time they're 18 or early 20s, they have lost hearing in one or both ears. The tumors characteristically form their bilateral vestibular schwannomas. Use my cursor here, I think you can see that. In the cartoon diagram, you can see the vestibular nerve, so the 8th cranial nerve. It's where these tumors, and just like the name sounds, they're schwannomas, so they're out of the Schwann cell lineage. And you, of course, see them with the arrows on the MRI. So desperate need in this patient population for targeted therapies to preserve their hearing. And there are other tumor types that I'm not going to talk deeply about, but they can affect mobility, obviously affect morbidity in these patients, and they cause a lot of significant amount of pain. And again, this is one of those slides, where people say, don't look at this too long. I just wanted to illustrate the protein that the NF2 gene encodes is called merlin. It's in the family, esrin, rhodixin, moesin. So it really interacts with the actin cytoskeleton and the cell membrane. So as you can imagine, it's connected to all that. You can probably see a lot of kinases in here, a lot of pathways. So again, the tumors are marked by the loss of heterozygosity of merlin. So what do? It's impacting so many pathways, interacting with receptors of the membrane, affecting so many things. So when we first got involved, when I was in Gary's lab, Wade contacted him, really interested in the kinome profiling and the potential to look in this rare disease. They're just desperate to find potential, and maybe we can target a kinase. So one of the initial studies in this consortium, funded by the Children's Tumor Foundation, was a plate screen of cell line models of schwannoma or meningioma, another tumor type that NF2 patients can be afflicted with, wild type, and NF2 null. And of course, a home run here would have been something that's selective for NF2 null, kind of representing if we could just kill the tumor cells, right? That's always our great hope. And this was done in collaboration with NCATS, a compound library, nearly 2,000 compounds were screened, follow-up synergy screens. And then from my perspective, working in Gary's lab, when they got to kind of some nominees or some candidate drugs or combinations, Wade had a mouse model, a genetically engineered model of NF2. And then at UNC, we were doing the omics, looking at the kinome, the transcriptome, and even other labs kind of looking at biochemical response, typically by Western blots, immunoblots. at the outcome for some of these drugs. And the top hits actually combinations for both of these cell line models of these tumor types included ALK-N1, an ALK inhibitor in combination with either an AKT inhibitor in the case of meningioma or dasatinib, again, approved for Ph positive, Philadelphia chromosome positive CML. ALK-N1 in their libraries, actually, they didn't have brigatinib, but ALK-N1 is an analog of brigatinib, an FDA-approved drug for ALK, anaplastic lymphoma kinase, which amplification seen driving lung cancer, neuroblastoma. And again, I should couch this, it was known even at that time from like kinome scan assays or other labs, like the Sorger lab and Dr. Mills had talked about, knew it hit other things, but still, oh, this is an ALK inhibitor. So we dug a little more deeply into that. First, the Wade's lab and another lab at Ohio State, the Chang lab, had a in vivo model, intracranial implant model of meningioma. They can monitor tumor growth through luminescence. And so at left, you're seeing what was interesting was compared to vehicle in blue and the growth over time by luminescence. Brigatinib by itself, which there's the green line, or at right in Wade Clapp's NF2 schwannoma model, brigatinib by itself kind of did the best. In the plot at left, you're seeing the DRG size, so dorsal root ganglia. The tumors form in a paraspinal fashion, so you can actually measure the volume of these along after dissecting the mouse. And I should say also, I'm not showing you here, but this also, the mice exhibit effect on their hearing. So just like patients have hearing loss, they can do ABR, test the hearing in these mice, and brigatinib also had a significant impact on that compared to vehicles. So it's really exciting. When we started to do the omics, just like has been introduced, the polypharmacology became clear. It was actually incredibly informative and valuable. So left, you're now looking at volcano plots of MIB-MS, kind of kinome profiling based data, where we're looking at compared to vehicle treatment, where do we see loss of binding after treatment with brigatinib? What kinases were being hit, if you allow me to say it that way, they're seeing loss of binding. So everything's significant, it shifted to the left. And so both in the meningioma cell line model treated for 24 hours, or in Wade's mouse model studying tissue, in this case, either for 3 days or 7 days, you can see some overlap, cyclin G-associated kinase, a P90, RPS6K3. But interestingly, we saw PTK2, a gene, it's the gene name, it encodes FAK, you're probably most familiar, focal adhesion kinase. In both cases, very substantial, very high degree of loss. And this actually, when we looked into things like kinome scan, other available resources, this was known. Other folks had worked with brigatinib and seen, oh yeah, by the way, it also hits FAK. And I should mention another lab, Joe Kissel's lab, in collaboration with Gary on another project, had seen crizotinib have an effect in NF2 tumors and went on through kinome profiling to show that, oh, this other ALK inhibitor, crizotinib, also hits FAK. And it's probably the important thing it's hitting. Now these, the cell lines got treated for 24 hours, the mice got treated for 3 days or 7 days. That's enough time for the kinome to rewire. There's a lot of plasticity there. And so we wanted to ensure that these were direct targets. So a couple of ways we can do that is just treat the cells for very short duration treatment, or add the inhibitor directly to the lysate. And that's something that the Kuster lab did and published on with a number of approved drugs to really define and understand polypharmacology. So in both cases, whether we did a short treatment, you're now looking at a scatter plot of 2 replicates of just drug versus DMSO at a few different concentrations. And again, you can see PTK2 is FAK1 in the cell line treated for a short duration. And in lysate from NF2 tumors from Wade's mouse model, we incubated with concentrations I've highlighted 10, 100, and 1,000 nanomolar. So achievable. The thing we're always interested in is, it's important, I think, to go to 10 micromolar. We're always interested in what we're seeing at kind of 1 micromolar, these achievable clinically relevant concentrations to get an idea what might be getting hit. So interestingly, there's no ALK expressed in either of these cell line models or the tumor tissue. So it's very clear that ALK was not a relevant target. But in this first portion, I just want to stop and summarize. NF2, quick example of kind of systems of biology approach, kind of nominating some drugs just by a pure cellular phenotype allowed us to move into, with collaborators, into these in vivo models. And the kinome profiling really, I think, like the way Dr. Mills said it, really helped us kind of deconvolve the polypharmacology. What targets are actually being hit? We know it's important to know what's everything that brigatinib could hit. And now in this very specific case, as she mentioned at the end of her talk, you kind of specific case reports in these NF2 tumors, what's it doing when we see a tumor reduction in our case. And then importantly and impactfully for the patient population, this led to the opening of the clinical trial that Scott Plotkin is leading. I think those results are, from what I'm hearing, a lot of a positive impact, not universal, but a lot of, so being able to repurpose this drug from ALK cancers into NF2 population. And the other case that I wanted to get is with NF1. We were working with Wade. Wade also has a mouse model that's kind of his expertise, getting these genetic models of these human diseases. And NF1, the nice thing is for ketchup, it has the same inheritance patterns in F2. It's about a 50-50 case, whether a parent, whether one parent passes along a mutation in one copy of the gene, in this case NF1, or it's sporadic in the germline. And then all the tumors and manifestations are listed here. I'm going to talk about tumors, specifically the neurofibromas from which they got their name. But again, it's marked by loss of heterozygosity. So you lose the other function of the other copy of NF1, this tumor suppressor. This is about 10 times more frequent, so it doesn't really classify as rare, rare-ish. But 1 in 3,000, in Indianapolis, I don't know, wherever, depending on where you are, like so the high schools here have 3,000 students. So you think about it in those terms, it's not as rare as often thought. As this audience well knows, NF1 is a RAS gap, a RAS GTPase activating protein. So when it's gone, RAS, the hydrolysis of RAS in this GTP-bound state is very, very slow. And so it's predominant in its active form. And patients, nearly 100%, it's actually not 100%, close to 100% patients have cutaneous neurofibromas, and 30 to 50% have plexiform. And as you can see in the MRI in the picture at right, these can be very, very large. They are still, that's a benign tumors, but they can become so large, they can compress vital structures. As you can imagine, they involve nerve fascicles, so extremely difficult to remove surgically. So if surgery and conventional chemo or radiation don't have an impact, they kind of just have to let them grow, causing a lot of pain and immobility for the patient, disfigurement, a lot of psychosocial burden, as you can see. And unlike NF2, where these tumors don't progress to malignancy, these plexiform neurofibromas are kind of a seedbed for transformation to a malignant sarcoma, malignant peripheral nerve sheath tumors, which are actually the leading cause of premature death for NF1 patients. And again, there's no approved targeted therapies. And if they can't be effectively removed by resection, by surgical resection, the patients have a very poor outcome. And again, in desperate needs of therapy, desperate need, of therapy. So the genetic events that occur, I told you these plexiform neurofibromas, this is C-bioportal data at the left, just from MPNST. The plexiforms are marked by loss of heterozygosity of NF1. Then they also lose, in the majority of cases, CDKN2A and CDKN2B. This 2A encodes two tumor suppressors, ARF and P16, and that really triggers transformation of these MPNSTs, which go on to lose components of polycomb repressive complex 2, which my lab is interested in, but I won't talk about today. So we kind of know, the stages and the genetic events that lead to transformation. But when we first started working with Wade and Stephen Rhodes, who was a fellow in Wade's lab, one of the questions we had is, let's just go all the way back to the starting point. Wade has a conditional mouse model of plexiform neurofibroma. Let's look at how these tissues differ from wild type nerve. And this really underscores when you lose NF1, you get GTP bound RAS, there's NF1 is gone. And actually it invokes, loss of NF1 gives you high RAS activity, and so you get markers of oncogene induced senescence. It actually upregulates the CDKN2A gene locus, giving you P16, giving you ARF, which of course leads to high P53. It's kind of a barrier, a senescent barrier to prevent further transformation. You can see senescence-associated beta-galactosidase staining in the mouse tissue and in patient plexiform neurofibroma. But what we did very early on is this is the lumbosacral region of microdissected from these mice, wild type or periostin Cree positive with floxed NF1, where they get these tumors. large nerve roots, large DRGs. We analyzed the tissue by MIB-MS and by RNA sequencing. You can see in this volcano plot, SARC is in there as well, but a couple top candidates. So this is just, can we use this to inform, give us ideas of what kinases might be targeted if they kind of go up, if we see an increase in binding in the tumors compared to a controlled normal nerve tissue, excuse me. And both of these are now being pursued, AXL inhibitors, or a marketed AXL inhibitor, multi-RTK inhibitor, and CDK4, CDK6 inhibitors. And this is now a couple of years old, but this really prompted further work in Wade's preclinical model. Again, shown at left, this is kinome data, cabozantinib treatment. This is a multi-RTK inhibitor approved for renal cancer and kind of maybe thought as more of a MET inhibitor. But we know it also hits AXL. It was efficacious in reducing tumor volume in the mouse model. You can see in the kinome plots near significant, this FDR of 5%. Hit some other targets that we're actually interested in, in the tumor tissue. And then at the right, this is the results of a phase 2 trial in a patient population in this waterfall plot, a 40% response rate. And really in an area, there is MEK inhibitor got approved, work in Wade's lab that, we were involved with when I was in Gary's lab and other clinical partners, was approved in 2020. And I think based on this, cabozantinib, there's still some more follow-up studies to be done, but it's looking promising for these patients as well in a kind of an area where they have selumetinib, they have a MEK inhibitor, really no other targeted therapeutic options for plexiform. And in the last couple of minutes, I'll just want to say that in Wade's lab, Stephen Rhodes published this paper kind of proving what's observed in the patient population. They knew conditional loss of NF1 in this mouse model in periostin-driven CRE could get plexiform neurofibromas, looked identical to patients. We also knocked out either the full CDKN2A locus encoding ARF and P16 or just ARF. That was sufficient to see progression to these aggressive malignant tumors, atypical neurofibromas, and then on to MPNSTs. So we have a model, a really tractable model. In mouse, we can study the tissue, but from my perspective, basic scientist, my background is all in cell culture and doing screens and cells to kind of give insight. So the beautiful thing about this is Lou Lei at UT Southwestern had developed this means, identified the tumor cell of origin, which from embryos at day 13 and a half, you can isolate these DRG nerve root neurosphere cells. They are Schwann cell precursors. It's kind of out of that lineage for NF1 also that these tumors arise. And then we can do the recombination in vitro with CRE, recombinase through adenovirus, confirm that, and then use those cells, reimplant them orthotopically for in vivo studies, do in vitro studies. And this gives us a cell line where we can do additional genetic perturbations. These are my last two slides. I'm wrapping up here. We've begun to do that and look at the kinome. I'm not going to get deeply into kinase targets yet. This is a little premature. We're kind of prepping this, but I'm comfortable showing you this. This is like cell of origin plexiform, and you can see increased binding versus control. You see CDK6 in there, which we observed before, but EphA2, a signature, even just, this is just in the kinases, a GO signature of neuron projection, some other insulin receptor, IGF-1R, and some CAM kinases there, and things we're following up on. And then if we knock out NF1 and ARF, I called it MPNST, but it's really, you could call it pre-MPNST. If we implant them, they will go on to form the malignant sarcomas. They've got the genetic hits. And so we want to look at what's the tumor cell of origin signature before we implant them. And we can see, again, CDK6 blew up even more in binding to the beads. We see that both at the level of activity, cyclin D goes up. We see it in increased binding by MIB-MS and at the level of RNA-seq. And you see a bunch of mitotic kinases, which makes sense. Now these are kind of unrestrained, fully transformed, capable of forming malignant tumors. So just wanted to summarize that. So we're taking these, multi-pronged approach. I think, definitely a high appreciation for the polypharmacology observed with any of these drugs, the need for, more selective inhibitors. And working is, I have the benefit of working with collaborators who have great mouse models. We can look at stages of tumor formation. And some of our ongoing work then with NCI collaborators and a lot of these trials is to follow up on, MEK inhibitor, selumetinib got approved. Probably other MEK inhibitors will follow shortly. There's some positive data with binimetinib and some others in this setting. and then cabozantin, a multi-RTK inhibitor. And so now we're able to get some of these patient samples pre and post. And they're benign tumors in these studies, so the adaptive responses are not as dramatic. The selection pressure isn't as high. There's typically a slow-growing tumor. And then also abemaciclib is of interest with the increase in CDK4, CDK6. And so we can do genetic validation to look at their role and kind of compare this to, CRISPR knockout with a genetic model compared to small molecule inhibitors. And again, just understanding where we need to go next. Like nothing so far has been a cure-all for all patients. So like with everything, we're going to be battling, individual patient heterogeneity and considering these adaptive responses. And the acknowledgements, a ton of names. I just need to acknowledge the folks involved with the NF2 work highlighted there in red. And then I have a second slide, apologies, but a number of names. And then now my lab at IU collaborate closely with Wade Clapp and Stephen Rhodes, their labs, and my NCI collaborators over at the right. We've got a U01 to look at the abemaciclib, some of these tumors that are capable of transforming. Thanks so much for your time.